Hello, and a very warm welcome to today's Cell and Gene Therapy Insights panel discussion, titled Advancing Cell Therapy: Innovations in Polymer-based Encapsulation and Delivery. I'm Abby Pinchbeck, an editor at BioInsights, and joining me today are a panel of experts who, among other things, will discuss the key characteristics of ultra-pure biopolymers, which can play a pivotal role in ensuring cell viability and controlled delivery of cell therapies. As always, do feel free to pose your questions to our panel using the Ask a Question box at the bottom of your screen, and we'll try to get to these during the session today. If you would like to direct a question to a specific panelist, please try to indicate that in your message. I'd also like to draw your attention to the Resources tab on the right of your screen, where you can find more information on the topics covered today. And so now I'd like to introduce your moderator for today. Dr. Joshua Katz is a Senior R&D Manager with IFF Pharma Solutions. He received his SB in Chemistry with a minor in Biomedical Engineering from MIT, where he worked under the guidance of Professor Darrell Irvine. From MIT, he moved to the Department of Bioengineering at the University of Pennsylvania, studying under Professors Jason Burdick and Daniel Hammer, and earning his PhD in 2011. He then joined the Formulation Science Group at Dow Chemical, where he worked on projects focused on encapsulation and controlled delivery for a variety of industries, including pharmaceuticals. As part of the Dow-DuPont merger and subsequent split, Josh joined the Pharma Solutions business, leading the colloids and biopharma R&D group within DuPont Nutrition and Biosciences. In early 2021, DuPont Nutrition and Biosciences merged with IFF, leading to his present position. His research today focuses on the fundamentals and applications of biopharmaceutical production and formulation. Josh has won several awards recognizing his research accomplishments, holds 4 US patents, and is an author of 15 patent applications, and has 35 peer-reviewed publications. So without any further ado, I'm going to hand over to Josh to introduce the rest of today's panel.
Thank you very much, Abby, for the kind introduction, and welcome everyone to today's webinar. We're pleased to have 4 esteemed panelists with us today. First, I'd like to introduce Dr. Evgeny Brudno. He is an associate professor in the Division of Pharmacoengineering and Molecular Pharmaceutics at the School of Pharmacy and the Department of Biomedical Engineering, jointly at the University of North Carolina in Chapel Hill and North Carolina State University. He earned dual degrees in chemistry and biophysics with minors in math and philosophy from the University of Pennsylvania, obtained his PhD in organic chemistry from Harvard University, developing directed evolution technologies with Professor David R. Liu. From there, he did postdoctoral research at the Wyss Institute at Harvard with Professor David Mooney, developing controlled-release drug delivery technologies for cancer and regenerative medicine. He joined UNC and NC State in 2017 with research interests that span organic, synthetic chemistry, materials science, and immunotherapy. Dr. Brudno's research is rooted in the belief that advances in chemistry and the basic molecular sciences can generate meaningful change in how therapies are designed, produced, and administered. Welcome, Evgeny. Dr. Marizella Della-Schlumbaum is a senior R&D manager at IFF's Pharma Solutions, leading a segment of the polymer synthesis and modification R&D team based in Baumwitz, Germany. Her primary responsibilities include polymer modifications, rheological characterization of synthesized materials, and technical leadership for the Novamatrix R&D platform. As an ultra-pure alginates expert, Marizella bridges the gap between biopharma technology needs and IFF's R&D and manufacturing capabilities, fostering additional opportunities in this field. She holds a Master of Science degree in chemistry from the University of Technology in Vienna, and a PhD in biotechnology from the University of Natural Resources and Life Sciences, Vienna. Welcome, Marizella. Dr. Tom Kemperman is an entrepreneurial biomedical engineer with a strong background in microfluidics, tissue engineering, and enabling microtechnologies. He has over 10 years of experience with the development of microfluidic systems for life science applications, and in particular, microencapsulation. Before starting IM Fluidics, he worked at various internationally recognized institutes, including the Max Planck Institute in Münster, Germany, and the Mesa Plus Institute for Nanotechnology in the Netherlands, the Technical Medical Center in the Netherlands, and the Division of Engineering and Medicine at Harvard Medical School. He is the recipient of the prestigious personal NWO Rubicon and Veni grants. In 2019, he was elected a European Innovator Under 35 by MIT Technology Review. Tom is the co-inventor of the in-air microfluidics technology, co-founder of IM Fluidics, and responsible for the technical management as IM Fluidics CTO. In this role, he has conceived, executed, and managed various innovative projects, including multiple large European consortia. Welcome, Tom. Finally, Dr. Omid Vaisa is an associate professor and CPRIT scholar in cancer research in the Department of Bioengineering at Rice University. He leads an interdisciplinary translational research program to engineer and commercialize next-generation cell-based therapeutics for various human diseases. His team leverages the latest techniques in synthetic biology, immunoengineering, and materials science to develop innovative cell-based platforms for real-time and feedback-regulated production of biologics. Over the course of his career, he has authored or co-authored more than 75 peer-reviewed publications, including those in Nature, Nature Biotechnology, Nature Materials, Nature Medicine, and Nature Biomedical Engineering, and is an inventor on more than 40 pending or awarded patents. He's also a serial entrepreneur who has co-founded Sigilon Therapeutics, Avenge Bio, Sentinel Bio, and Curata Bio. These companies collectively have attracted approximately $500 million in private and public investment capital. In September of 2003, he was named the director of Rice University's Biotech Launchpad, a new initiative with a mission to accelerate the translation of Rice University discoveries and technologies into clinical practice to provide rapid patient access to leading-edge therapeutic proteins. Welcome, Omid. Perhaps to kick off the session today, I would invite each of you to please give a brief overview of your work, especially as it relates to cell encapsulation and delivery techniques. We'll start, please, with Yevgeny.
Sure. Hey, thanks, Josh, and great to join you for this panel. So work in our lab, I think, centers on manipulating immune cells, so right now mostly T cells, with biomaterials and using biomaterials principles to make therapeutic cells, make them faster, cheaper, and make them more potent and deliver them in new and innovative ways. And one thing that we're really focused right now, which I think is of particular excitement, is a material that can make CAR T cells in a couple of hours. So CAR T cells take about 4 weeks to make, but we've sort of created materials that can do the same process in a couple of hours and be implanted for cell delivery. And so we really think about how material science can influence these kinds of expensive and difficult therapies.
Thank you. Omid, your thoughts? Omid, you appear to be muted.
Oh, can you hear me now?
Yes.
Okay. Yeah, my lab is interested in developing cell-based therapeutics. These modalities are really engineered cells that can produce biologics in the patient. When we think about the pharmaceutical industry today, most of the drugs that are having big impacts on patients' lives are biologics in nature. And these biologics are made in steel bioreactors around the world. Radial loss and logistics goes in purifying, isolating, stabilizing, and ultimately the patient has to administer these through injections or infusions. What we like to do is think about a new paradigm whereby the biologics are produced in the patient's own body, really reducing the logistics burden, reducing the cost, and improving patient outcomes and enabling certain therapeutics that are not possible today. So we leverage cell encapsulation as a way to protect allogeneic cells from the host immune response. Spend a lot of time thinking about the access of the biomaterials and how to modulate it to where it can be stealth to the body. So that's a good deal of what my lab is focused on.
Thank you very much. Marizella, can you talk a little bit about an overview of your work, please?
Yes, sure. As you've already mentioned, I'm part of the polymer design group here in Bomlitz, and our core business is really modification and fine-tuning of polymer derivatives and functions, structure, properties, and also the interaction with different cores, like, for example, cells. So my main work is understanding the gaps on the market and trying to find a solution in the lab that can be used in different applications, for example, for encapsulation of cells.
Nice. And finally, Tom, you'd like to give an introduction to your work.
Yeah, thanks, and nice to meet you all. I have a background in biomedical engineering, and I worked a lot on single stem cell encapsulation using droplet microfluidic systems. And already quite early during my work, I experienced also the pain of microfluidic systems. Well, actually, it has a lot of advantages, but it's also pretty slow, and you have a lot of clogging issues. And then we started to work on what we call in-air microfluidics. It's a microfluidic system, but then off-chip by combining liquid jets in mid-air. And using that system, actually managed to upscale microfluidic cell encapsulation process. I've used that for several applications, amongst others, tissue engineering applications to create new types of capsules at a rapid pace for to study cell material interactions, but also, for example, to protect cells during 3D printing applications. And still do that now as a CTO of IM Fluidics, where we try to commercialize this technology.
Great, thank you. Maybe I'll stick with you. You mentioned the beginning of what you said there about some of the challenges you faced early on with microfluidics. Could you talk a little bit about what you consider to be the main challenges relating to materials being used in cell encapsulation applications today?
Yes, of course. Yeah. So I think there are several challenges, but for me, the big challenge is combining several small challenges into one solution. So typically, to have a successful solution for cell encapsulation, you need to integrate biocompatibility or cytocompatibility, if you will, with the functional material. It has to be scalable and at the same time compliant with regulatory. And very often the most interesting materials that are out there are only available as in small amounts, and they are niche products or even niche research projects that are existing but not scaled yet and not available off the shelf. So I think if that to be able to test at scale with off-the-shelf components is still a big challenge. In itself, scaling the encapsulation process is a challenge, because you want to have the resolution of a microfluidic process, or at least you want to have single cell resolution to encapsulate something. But doing that at scale is tricky, because then you start to use, for example, small systems that are prone to clogging and are difficult to make using clean room technologies. So we need kind of new micro-manufacturing methods to be able to scale microencapsulation, I think. Another challenge, I think, as a result of this is the limitation of scaling 3D cell culture and cell therapies. So if we can find solutions to upscale cell encapsulation with the right quality and resolution down to the single cell level, then we can also enable new cell therapies and maybe cell preservation methods as alternatives to the current cryopreservation strategies.
Great, thank you. Omid, maybe I'll kick it over to you and ask you your thoughts on what the main challenges are today as related to cell encapsulation.
So, yeah, I would say, there's two buckets. One is on the cell engineering, the synthetic biology, creating cell lines that are potent to enable therapeutic applications. And then on the material side, it's really identifying materials that can interact with the host immune system in predictable ways. So those are probably the two major areas of thrust: is how do we find better and better materials for encapsulation? And then on the opposite end, the cells themselves, how do we improve their performance, their viability, their potency, their clinical translatability?
Very good, thank you. And Yevgeny, perhaps your thoughts on the main challenges. Appears as though you're muted at the moment, Yevgeny. Okay, maybe while you try to figure out unmuting yourself, I'll kick it over to you, Marizella, instead. Can you talk a little bit about what you consider to be the main challenges relating to cell encapsulation applications?
So I think it starts with choosing the right polymer. So there are so many options. There are natural biopolymers, there are synthetic polymers, there are animal-derived polymers, non-animal-derived polymers, all of them with different properties and different advantages and disadvantages. This comes along with the purity and availability, as Thomas already mentioned, accessibility in larger quantities. So all of them are not also compatible with every single technology or method that is currently used for encapsulation of cells. Then, of course, when you've chosen the material, you need to ensure that the materials do not trigger any immune response in humans. So natural polymers are generally well tolerated, but also there are lots of differences. I think also regarding different technologies used for encapsulation, the material should possess mechanical resistance so that it still protects the cells during the procedure of encapsulation. There are different technologies. Some of them require shear thinning, some of them require low viscosity, some require high viscosity. So all this should be enabled by the choice of the right polymer. Of course, when you think about already encapsulated cells, the material should enable some permeability of oxygen and nutrients. So they should be all able to diffuse through the materials or diffuse out of the material of the capsule. And the material should also enable that the cell stays vital and viable during the period of encapsulation. Sometimes it's just a few hours, but sometimes it's days or weeks that require the cell to survive in an encapsulated status. So I think purity is also a big topic when we talk about encapsulation of human cells. As has already been mentioned, really also the variability within batches can also be a challenge.
Great, thank you. Yevgeny, I believe
Yeah.
so maybe if you could share a little bit, what you see as the main challenges, but then also maybe we'll move on as well. With the main challenges, what do you also see as some of the most promising approaches to address these challenges?
Yeah, so thanks. Good question. The thing that for us has been a really challenge is, I think, from a chemistry or a molecular point of view, the scope of what we can do is nearly infinite, right? We can modify materials and display molecules in almost any way you can imagine, but understanding what we should be doing and understanding how chemical changes that we might make or material choices that we might make will influence cells, especially in the long term and especially inside of patients or inside of sort of in an in vivo setting, has been just incredibly challenging, I think, for us and for the field. And one thing that we focused a lot on is kind of moving into in vivo settings, including potentially NHP settings, as quickly as possible, because something we found is cells and in vitro settings are just not predictive of what will happen or how a material will truly behave or how a material will influence cells. And so it's sort of an ongoing problem of how do you make smart decisions on a chemical or a molecular level, to have the kind of influence that you're hoping to have therapeutically.
Great. And so what would you say perhaps some of the most promising approaches then are to address these challenges?
I actually think the most promising approaches have nothing to do with material science. I think, bringing in really novel imaging applications, really novel ways of understanding what a cell is doing in vivo and whether it's happy or not. Those are sort of the things that we're really interested in, and that those are the things that I'm most excited about right now.
Okay. So Marizella, Yevgeny says that it's not a materials approach. You're a materials scientist, lead a materials development group. What are your thoughts on those promising approaches? What do you have to say in response?
I do think that there are many more challenges than just with the materials that are used. I totally agree. This field is so complex and so big. The used material is just a really tiny piece of it. But I still think that there can be done some improvements to increase the performance in a system. And I do think that smart polymers or biopolymers with high purity will get the chance to get a bigger attraction in this field once we find a way to mimic the extracellular matrix environment by using non-animal derived materials. So I think a lot of people are working on that, and we belong to those, of course. I'm of course pro non-animal derived materials, and I do think that this is a very interesting field. In my area, we really try to understand what every change in the polymer structure causes to its function and what does it cause in a certain application. This area, encapsulation of cells, is just a technique, but the application can be so diverse. So also serving the applications is very important to understand what are the needs in a certain application. Sometimes it can be delivery of cells in humans. Sometimes it can be a long-term survival of cells during a transportation in a capsule. Sometimes it is growth of cells during a cultivation period. So all of these require different properties and have different challenges. So I think offering specific solutions for a specific area may change a little bit the field and may close some gaps, but I fully agree there is so much more to the side of the material science.
Tom, what are your thoughts on some of the most promising approaches to address the various challenges we've discussed?
I think, yeah, first of all, I agree with what's being said. And then at the same time, I also see the need to be able to scale all of this to something clinically and industrially relevant. Because if we can only do it in the lab, then it's not of use. So I'm always looking for ways to upscale kind of the combination of cells and materials, or cell only also fine. And that scaling needs to be solved. And I think that there is a big opportunity for new processes, especially to enable suspension culture in bioreactors of cells and 3D cell aggregates. So it's not trivial to culture 3D cell aggregates in a bioreactor, because they start to merge and they start to form, let's say, multi-spheroids or multi-organoids, and then you lose kind of the level of control that you want in your system. And cell encapsulation, I believe, can play a very important role there to protect cell aggregates and organoids from, first of all, merging, and second of all, from the shear forces that are typically present in bioreactors. And well, related to the imaging part, I think the cell capsules can also provide a nice 3D microenvironment that kind of preserves the cell 3D cell shape while imaging. So it enables you to, outside the body and outside the culture or petri dish, still do representative 3D imaging within an encapsulated system. So there I see a lot of interest, and or I have a lot of interest, and especially finding ways to enable that scaled encapsulation using new approaches. And particularly, I'm interested in injetting approaches, because I think that's intrinsically faster than the common dripping approaches that we see in the field.
Great, thank you. And Omid, your thoughts on some of those promising approaches?
Yeah, I mean, I think the big, push for all of us is to advance some of these technologies to the clinic to create a marketplace, and then that can kind of create more feedback loops. But yeah, I mean, I think I am excited for a future where we have cell therapies, approved products, because that would really create a bigger incentive and opportunities to innovate in this field.
Great. Thank you. All right. Evgeny, I'm gonna ask you now. So you mentioned that you're doing some work with T cell therapies, and today commercial T cell therapies, as you mentioned, are modified ex vivo and ultimately administered as suspensions. And generally, T cell therapies, at least today, are limited in their efficacy to liquid cancers. So, how could novel technologies support the continued improvement of T cell therapies to more challenging targets such as solid cancers?
Yeah, I think biomaterials have a really large role to play here, and I think we if we take a step back and sort of ask why solid tumors haven't been as successful, I think you can focus on a couple of areas: T cell penetrance into tumors, as well as immunosuppression of the T cells within a tumor environment, are probably key areas. And I think biomaterials have a lot of promise in overcoming those issues, whether it's delivery from biomaterials into tumors to enhance the penetrance, or the well-known role of biomaterials to enhance immunogenicity, or especially of delivering some immunoactive factors or sort of immunostimulating factors. So I think there's a nice role to here. As an added thing, I would add, and this is something that we're very actively working on in our lab, is the ability of biomaterials to recapitulate nurturing niches for T cells, right? So generally T cells are sort of exist in a nutrient-rich lymphoid environment before they have to go and fight a tumor, or go and fight disease. And if we're just IV administering, uh, we're skipping that part, right? And so the ability to create an in vivo nurturing niche, an in vivo microenvironment that can stimulate cells in just the right way, I think is going to be really important to create cells of the quality, lack of exhaustion, the right differentiation state that will make a difference for sort of new treatments that are currently out of reach.
Great. Thank you. So, Tom, coming from the encapsulation process development side of things, and Marizella coming from the polymer design side of things, I really want to ask both of you what you consider to be some of the key characteristics you consider during the design of a new encapsulation and polymer technology, respectively. Maybe, Tom, we'll start with you. Talk about the design of new encapsulation technologies, what the key characteristics are that you're looking for.
Yeah, sure. Yeah, thanks. I already mentioned a little bit. I think it's at least 4 things that are important. It's resolution, biocompatibility or cytocompatibility, the flexibility of the process, and scalability of the process. Let's start with the first one. Encapsulation process should be compatible with processing liquids at the resolution of interest, which I think is related to what you want to encapsulate. For example, a single cell or a spheroid or organoid, and the process has to match that resolution, because it has been widely demonstrated that if the constructs that you produce are too large, you create necrotic core formation, and that's unwanted, or you get an uncontrolled condition where you get gradients of growth factors over the construct that cause different outcomes of the experiment. Biocompatibility is very important, and specifically related to shear forces and temperature and processing solvents. And that's not trivial. That sounds trivial, but with conventional chip-based microfluidics, for example, it's very common to use emulsification in combination with oils and emulsifiers. And we know that those can be harmful to the cells, or it's at least unwanted for follow-up cell therapy, where you want to work as clean as possible. In terms of flexibility, I think it's very important to not be in the way of the formulation chemist. You want the formulator or the chemist to be as flexible and free as possible to design whatever he or she wants. And very often liquid processing stands in the way because it's limiting in terms of viscosity that can handle, or maybe in terms of again, nozzle size causing clogging, etc. So you want to be as flexible as possible. And in the end, you want to be as scalable as possible. And that's also not trivial, since the most high resolution liquid processing methods contain very small through holes that give a lot of hydrodynamic resistance and therefore are very slow in throughput. So you need to parallelize your system or find other ways to overcome the inherent slow throughput of high resolution systems. And that is that is what I like to focus on. And typically the answer there is to combine multiple technologies. So one thing that we did specifically is combine a vibrating nozzle technology that operates in the jetting regime, combine it with a microfluidics approach where you combine different jets on the fly, and thereby you have the advantage of something fast and clean and mild, combined with the flexibility and inline control of a microfluidic process. That's how you could look at that.
Great, thank you. And Marizella, can you offer some additional thoughts? I know we've spoken to some of this already, but some of the key characteristics around polymer design that you look for some emerging encapsulation technologies.
So when we look specifically at the encapsulation of cells, there are two different types of cells that mainly come into focus when we talk about cell therapies. It can be adherent cells or non-adherent cells. So when we talk about encapsulating adherent cells, we try to add some function to our polymers so that the adherent cells can really attach to the polymer. Alginate normally doesn't have any recognition sequence for adherent cells, so we try to mimic the extracellular matrix environment of the natural environment of the cell by adding short peptide sequences to our polymers to enable the cells to attach to those, to recognize them and attach. So these are the things that we can do. So we try to fine-tune the polymer so that it really has its best performance in the application. Thomas already mentioned a lot about the mechanical properties. So when we think about a hydrogel that is formed as a shell around a cell, it really needs to be as soft as natural tissue. So the polymer can, for example, form very viscous solutions, which can make the processability very difficult. So shear thinning is, I think, really, really important for many technologies that are currently available for encapsulation of cells. And the purity of the material, I think, is key, because also small impurities can clog the needles and nozzles, and all of this can be avoided if we just start with very highly pure materials. I think Thomas covered already a lot that were also my thoughts, and I really think also the biocompatibility and the compatibility of the chosen material with the certain cell line that you want to work with is very important. I think these are my additional thoughts.
Great, thank you. So, Omid, I'm gonna come over to you now. You obviously have significant experience not only as an academic researcher, but also, in your words, as a serial entrepreneur, having started companies. Some of these companies have since been acquired by large pharmaceutical companies. So could you discuss some of the similarities and differences in the role of research and development from academia to startup companies to large pharma companies, especially as it relates to cell encapsulation, right? What role does each one play? How's it different? How's it similar? Your perspective and some of your experience there.
Yeah, so I think in the lab we can iterate quite effectively with a diversity of different strategies to consider. But then as you start companies and get acquired by bigger companies, manufacturing and scale up become bigger issues, and coming up with processes that are scalable, I think, is critical. And that's something that we think about the earliest stages of our research, because our program is really focused on product development. And so, it's critical to think about things like, how do you develop assays to validate the stability, sterility, purity, potency, viability of the cells, encapsulation material. How do you QC that it's the same every time? How do you manufacture in a way where, in the lab we're always making like maybe milliliter scale materials for testing, but then when you do a phase 1 clinical trial, that really has to be expanded to liters, and ultimately you need to be able to scale this to an operation where you could serve the masses, patients in some cases. Indications could be quite large, hundreds of thousands of patients just in the US alone. And so you really have to think about if what you're thinking, what you're developing in the lab can eventually mature to that level. And that's hard to do, but you have to, think about keeping it simple enough to where it's possible. And then sourcing the materials so that they're available, and finding good partners, which, like DuPont, they've been great to work with over the years.
Great. So I'm going to stay with you. So there's a lot of work going on with T-cell delivery and pancreatic islets, with cell encapsulation emerging as a new delivery technique. What specific therapeutic areas or indications do you see as being most relevant?
Yeah, I would say, this field really got started with the work with pancreatic islets, thinking about some of the pioneering work that Lemon-Sun did in 1980. It was a science paper where they encapsulated rat islets and showed that you could reverse type 1 diabetes in these animal models. That really spurred a big flurry of activities over the years. Many clinical trials launched. Obviously that concept is still compelling. I would say that that's probably one of the more difficult concepts because cells to both sense and respond and secrete insulin. Islets are particularly potent cells. I think what's emerging today is really unique opportunities to leverage advances in synthetic biology where you can engineer other cell chassis for biologic production and then use these as therapeutics where the biologic is produced in the patient. There's a lot of different indications that this makes sense for, what ranging from inflammatory conditions where antibodies could be made, oncology. We created Avenge Bio, which is an immuno-oncology company that advanced products to the clinic, where cytokines are being delivered to provoke an immune response. There's a lot of opportunities with type 2 diabetes and metabolic disorders, producing hormones that can regulate insulin production, obesity, weight loss. So I think there's a lot of opportunities for with other cell chassis. And in the context of T-cell delivery, there's opportunities to do that in vivo manufacturing, like Yeti and his team has shown. But also others are looking at how do you encapsulate ex vivo manufactured T cells and use those to spatially constrict and deliver them to tumor beds. And that's also an emerging opportunity for sure.
Great, thanks. Danny, I want to see if you have thoughts on the same. Obviously you're working in this space as well. So you've been doing a lot of work around T cells. You want to talk about some emerging areas or therapeutic areas or indications that you see as particularly relevant?
Yeah, thanks. I think Omid really nicely and succinctly put it. I think the really emerging areas are going to be ones where right now there's just very few other options, right? Things where we've been trying for decades with small molecules or trying for years with biologics, and we just have not been successful. And we really feel like we need really new thinking and new approaches, and full cellular therapeutics. And clearly, I think cancer is an area where it is clear that we just need additional tools in the toolbox to have the kind of impact we're hoping for, right? Really give patients years or decades of life. Diabetes, where we have not figured out a way to just control this disease permanently and without interventions for years or decades. And I think those kinds of areas are always going to be the most exciting and most relevant therapeutic indications. But as biomaterials and encapsulations and cell therapies catch on, I think as the costs go down, we will access therapeutic areas that are maybe manageable with current areas, just in a way that's suboptimal. But things where people are dying, I think those are always going to be the most relevant and most exciting therapeutic areas.
So, Tom, in Omid's answer just now, one of the things he talked about was the ability to go from small lab scale, typical of what is seen in an academic lab, to being sure you can have material for clinical trials, larger patient populations, larger volumes. So, can you talk a little bit about how we ensure scalability and quality control and reproducibility in a polymer-based encapsulation system?
Yeah, of course. Well, it will be a little bit of a generic answer, and it's also, well, if you have the answer to all of this, then yeah, would not be here. But I think one of the well, what I like to do is at least try to work with off-the-shelf components as much as possible. But this is not always possible, because then the solution will not be innovative anymore. And wherever you need to iterate and do kind of fundamental work, I think it's at least smart to do it in a modular fashion, where you kind of isolate that unique new material or a cell or component, and treat it as a modular component, as part of the total system. So you can also easily replace it at a later stage, or you can do concurrent engineering with multiple teams to solve multiple problems at the same time. That is kind of the way, I think, to approach it, to also make it the solution scalable and efficient. One of the other things that we did not discuss that I'm interested in is scaling of cell culture, not only by encapsulating in 3D, but we can also use these biomaterials for 2D or 2.5D cell culture using microcarriers. And I think microcarriers are still a great solution to scale up cell culture in bioreactors. There are advantages and disadvantages, but I think there is at least one shortcoming, that there's only a handful of microcarriers, and there are a dozen different cell types, and there is a mismatch. So if you want to scale cell production or cell expansion, it will also help to have more custom or customizable microcarriers. That can really be that can be mixed and matched to the cell type you want to expand.
Great, thank you. Marizella, we see a lot of excitement around the use of alginates for cell encapsulation, an area I know that you do quite a bit of work. Can you talk about some of the unique features that make alginates so attractive for this space?
Yes, definitely. So alginate is biocompatible and a non-toxic polymer, so it's perfect for interactions with living cells, and that's one of the great properties. Another is probably the easy and very mild gelation. So alginate forms a gel under physiological conditions by just cross-linking with a divalent cation. So the gelation is independent from temperature, pH, and can be done at the ambient temperature and pH that are normally used in cell culture applications. This makes it very attractive for applications with living cells, where mild gelation is really a requirement. And this gelation is easily reversible. So you just need to take away the divalent cation, and the entire hydrogel will dissolve. Alginate, besides all of this, allows also co-encapsulation of cells with other biomolecules, for example, with growth factors or drugs within the same microcapsule. There are lots of studies about that. This can even enhance therapeutic potential. And it can be combined with other polymers to fine-tune its mechanical properties. And we see this a lot in 3D bioprinting, for example.
Great, thank you. I do want to encourage questions from the audience as well. So those of you who are joining us live today, if there are questions that you'd like to submit, please feel free to do so. And if they're for a specific individual, please indicate so. Otherwise, I'm happy to see the questions and spread them around as appropriate. Want to make sure that we're certainly addressing the topics that you, the audience, are interested in as well. We do have a question that's been submitted so far, and so I'm going to ask Tom for you to take this one. The question is, I think, building off what's been done with the lipid nanoparticles for COVID-19 vaccines. So a question around the encapsulation process and trace residuals. The question specific around formaldehyde, but maybe we can take it beyond that. You certainly mentioned, microfluidic processes that use co-solvents and emulsifiers and the like. Can you talk a little bit more about trace residuals that come out from various encapsulation technologies and any implications that might have on clinical performance?
Yeah, first of all, if you don't need it, don't use it. That's the easiest, because if you put it in, you have to get it out, and you have to demonstrate that it's out, and that's not easy. If you really need it, then yeah, you have to find validated methods to prove that the residual material is no longer there after production. So it's, I think, as Marisela also mentioned, there is a benefit of alginate here, because that's the cross-linking of alginate can be done very mildly without any chemical cross-linkers. The downside is that it's then only physically cross-linked. So there is pros and cons. So maybe the network is less strong. But if you really want to chemically cross-link something, then formaldehyde is used a lot. And there are already a lot of assays that can demonstrate the activity of the formaldehyde after being used. I'm not really familiar with then the final regulation or the final assay that you have to show in, for example, a clinical trial, or in a product. Of course, you have the general toxicity analysis, etc. But there are pretty good ways to demonstrate any leftovers of particularly the formaldehyde, and as well as neutralizing it after using it as a cross-linker. So maybe one of the other speakers or panelists can also weigh in if they have more experience with this.
Anyone have anything that they would like to add on, residuals or traces or impurities that might be present?
Yeah, I mean, I think it's a really good point and a very challenging thing. So I agree. If you don't need it, don't use it, because it's really hard to get it out, and the FDA does require you to do residual solvent analysis, and really the levels that are acceptable are extremely low, and so it is challenging. Great.
We have a questions also come in, or a few questions around alginates specifically. One around some of the key features that make alginates specifically biocompatible. And Marisela, maybe you'll start us off with that. But then for the broader audience as well, there's some questions around alginate conjugation with peptides and what's been done with peptides. And I think several of you have some experience working with alginates coupled to peptides and other functional groups. So we'll pass that one around. But maybe, Marisela, if you'd like to get us started specifically on what makes alginate so biocompatible.
I mean, it's a natural biopolymer, and it has been in use with cells for multiple decades. So it has been studied for many, many years, and it is well known to not have any harmful reactions to cells. So the cells can really grow, proliferate, and survive within an alginate hydrogel. This is what is understood under biocompatibility. So it has no negative influence on the living cell. On the question about the peptide coupling, so we use sequences that are known to be recognized by cells integrins. by adherent cells. So this is a natural mechanism of a cell that triggers attachment to certain sequences within that are known to be present in collagen or any other extracellular matrix protein. And we mimic this process by reducing this sequence to really its essence. Mostly it's 3 to 6 peptides long. And we use this to mimic this extracellular environment to enhance the attachment of cell to a certain material, because adherent cells need to attach to something. If they do not attach to a material, they will attach to each other, and at some point form clumps and spheres, organoids, spheroids, or whatever, and at some point die. So this is what we try to do with the coupling of peptides to alginates.
There are others who have experience with coupling peptides are similar to alginates and want to share a little bit on their experience with doing that.
Yeah, I can add a few words. Our lab has done quite a bit of sort of peptide alginate conjugations. It's one of the reasons we like alginate, that the conjugations are straightforward and chemically very easy, and it's sort of a very facile reaction to do. So there's a kind of a pretty established body of literature on modifying alginates with various collagen mimicking peptides, integrin mimicking peptides that just benefit cells to spread out and bind and to be used in things like tissue engineering applications.
Great. Looking at the next question that came in, and maybe Omid, we'll start with you on this one. There's a question around cell viability over time. So cells that are going to persist encapsulated in an alginate matrix for a long period of time. Specifically, the question is asking about adherent cells. So if you have thoughts on adherent cells, but also more broadly, cell viability over time. What do you see? What impacts the cell viability over time? How do we enable these technologies to last for long periods of time?
Yeah, so it depends on if it's in vitro or in vivo. I mean, I think in vitro, really, the nice thing about alginate is that its cross-linking scheme is a gentle chelation mechanism, which does not cause any harm to the cells. Other materials that people have used for encapsulation, where kind of covalent reactions are needed, those can create byproducts that can be toxic to the cells. But with alginate, the nice thing about it is it's porous enough to enable diffusion of key molecules. Need nutrients to get in, but cells also are very dependent on things like transferrin. So transferrin is one of the molecules that exists in cell culture media that are produced by cells, and then the T cells can benefit from it. But it's kind of a bulky molecule. It's about 100 kilodalton, so it has to be able to penetrate through. And the pore size of alginate is nice because it allows for these molecules to get in and out, but immune cells can't infiltrate in. In vivo, of course, the barrier becomes foreign body responses, which any material you implant in the body creates that, and that can be a barrier to long-term viability. We've gotten around that through the whole modification schemes to prevent foreign body responses, and that can help. But ultimately, it's a combination of super biocompatibility to prevent foreign body responses and ensuring that the cells get consistent and durable access to nutrients and oxygen. And those are the key criteria to the long-term performance.
Great. Anyone have
Maybe I
others?
also
please.
would like to weigh in. I fully agree that the chemical part is very important, and that alginate and in general hydrogels are semi-permeable networks that with the right molecular weight cutoff they can support let's say cell function because of the diffusion of the nutrients and waste and growth factor. The other important aspect here, I think, is the mechanical stimulation. Cells do need to have some mechanical input, otherwise they will go quiescent. They will go into sleep mode and eventually may even die. And this is especially the case if you do single cell encapsulation or basically single cells throughout a bulk hydrogel or in microcapsules. Then it's very important that cell can actually feel something, that they can feel their microenvironment, either by depositing their own pericellular matrix or by directly interacting with the hydrogel. And also there, the peptides, especially peptide modified alginates, can play a big role, because pristine alginate does not interact with cells. So there is no push and pull, and cells will indeed die or at least go quiescent if you don't present any cell binding moieties. For cell spheroids, that's different, because then they have each other. So they feel something and they are basically usually doing well. But what's also important, that cells respond to mechanical input, especially stem cells are known to differentiate also based on mechanical input. So in a soft environment, they are more prone to adipogenic differentiation, or the softer tissues, whereas in a stiff environment, they can differentiate or are more prone to differentiate towards stiffer tissues, let's say bone tissue or cartilage. And there, the material cell interaction is super important and can be mediated by, for example, cell adherent peptides.
So we only have a few minutes left. So, perhaps, we'll move to the wrap-up question. And it's really, about, for each of you, what one word or phrase do you see as the most promising novelty in this return field? We've covered a lot today. There's a lot of promise. There's still a lot of challenges. It's a rapidly growing space. So, just to sum it up really quickly for each of you, what do you see is most promising? What has you the most excited right now? And Tom, I see you on screen right now, so why don't we start with you?
Yeah, okay, thanks. Very short, it's novel microfluidic technologies to enable scaling of cell culture. And in one word, I'm very biased, it would be in-air microfluidics, because that's what I developed, but in general, microfluidic systems to scale cell encapsulation.
Great, thank you. Evgeny, what are your thoughts?
Sure, I'll be biased too. I'm going to say bioinstruction, right? The ability to tell cells what to do and how to function inside the body in complex environments.
Omid? You're muted, Omid. Never. You muted again. Omid, you need to unmute.
Oh, I thought I did. Can you guys hear me now?
Yeah, hear you now. Great.
So yeah, for me, it's really the unique opportunity to modulate host immune responses using the material design. I think that interplay between the design of the material and the immune reactions, the modulation that can do, is quite exciting. And the more we learn about immunology and immunoengineering, really creates unique opportunities to hack into the immune system and do some innovative things that could be very beneficial to patients.
Thank you. Marizella, your closing thoughts?
Well, I would say combination of smart polymers like alginate with new smart technologies to encapsulate cells may be beneficial.
Great. Well, I want to thank all four of you for joining us today and being part of this panel. I thought it was a very interesting discussion. We learned a lot. And Abby, I'd like to pass it back over to you for some closing comments before we close out the webinar. Thank you, everyone, for joining today.
Thank you so much, Josh. And thank you to all our panelists for a very informative and thought-provoking discussion today. And thank you to Josh for doing a fantastic job moderating today's session. Unfortunately, that's all we have time for, but any questions that we didn't manage to get to, we will reply via email. Today's webinar will be available on demand tomorrow, so look out for an email from us with the link. And all that's left is to thank all our panelists once more for a great discussion today, and thank you to the audience for listening. We hope you'll join us again soon.
Thanks, Abigail, and thanks for organizing.